Hello everyone, my name is Dr. Avelyno D Costa, and in this video I will be talking about the different types of DNA damage in continuation from my previous video where I also spoke about some other types of DNA damage. In the previous video I spoke about the different types of point mutations, then DNA alkylation, deamination, hydroxylation, DNA intercalation, etc. And in this video I'll be talking about some other types of DNA damage. So to begin with, we talk about the adducts or the adduct formation. So there are some compounds which are present which can bind covalently with DNA to form a very large complex structure which is referred to as an adduct. We refer to these structures sometimes as DNA adducts or bulky adducts, and certain compounds are there, like the example that is given here is that of benzo alpha pyrene. So benzo alpha pyrene is a compound that is found in petroleum products. The number of compounds which are found in petroleum products, one important one is benzo alpha pyrene because it is a highly toxic compound that is found. So benzoalpha-pyrene is a known DNA damaging agent in the sense that it can bind to the bases of DNA, like guanine, and it can form a very large structure that looks like this. So this adduct, when it forms, it is going to cause problems in the structure of the DNA helix. It will lead to distortion of the DNA, and obviously it will lead to then further problems of, if the DNA has to undergo replication or transcription, this particular adduct will not be able to be recognized properly as a base, and probably then replication would not happen or transcription may not happen at that point of time. So adducts can also lead to another type of a damage which is referred to as DNA cross-linking. So you can see when an adduct binds to DNA, it forms a bulky structure that looks like this. It is referred to as a monoadduct. But some adducts are there which can actually cross-link or connect two bases within DNA. Like for example, there are some compounds which can cause interstrand cross-link, where a base of one strand via the adduct can form a link with a base on another strand on the other strand of the DNA. Okay, so you can see that this is referred to as interstrand cross linking. So this forms a sort of like a bond between a base on one strand and a base on an other strand. Again, this will lead to distortion of the DNA helix and problems in replication and transcription. The other type of crosslink, as you can see over here, is an intra-strand crosslink, which is the crosslink that is formed via the adduct between a base on one strand and a base on the same strand. So within the same strand, there will be an intra-strand crosslink which takes place, a bond formation with via the adduct, and that can also lead to problems of transcription and replication. And another type of cross-link you can see over here is that some adducts can actually form a link between the base of the DNA and a protein that is going to probably be found in the nucleus. And when this happens, again, this will lead to a very large protein that is going to be bound covalently like this to the base, which is not supposed to be there. There are a lot of proteins which are in close association with DNA, like histone proteins and the scaffolding proteins, but they don't alter the structure of the DNA as such. That is, they don't permanently, they don't they don't distort the DNA helical structure. Whereas this can actually distort the DNA helix. It can lead to interference of transcription and replication as well. The next type of DNA damage is another important one, which is the oxidative damage. Oxidative damage is brought about by some compounds or some molecules which are referred to as reactive oxygen species or free radicals. And you can see that there are different types of free radicals or reactive oxygen species which occur in our body. These can be produced under normal circumstances in the process of cellular respiration, but these are going to be quickly detoxified by the body via the antioxidants and the antioxidant enzymes that are there in our bodies. The problem arises is when these free radicals increase to such an extent or to such a concentration that it overwhelms our antioxidant systems, and many times and often it actually it, such conditions can occur where the levels of reactive oxygen species in our body far exceed the concentrations of the antioxidants that are there in our body. So what do ROS do, or what do they are abbreviated as ROS, reactive oxygen species? These ROS can actually damage different cellular structures. That is, they can damage DNA, they can damage proteins, and they can damage lipids. So they are known to be cytotoxic in nature, and oxidative damage, it can lead to, or if it damages the DNA, then it can lead to the DNA not being able to replicate properly. It would not be able to be transcribing, and so definitely it is a DNA damaging agent. And oxidative damage has also been implicated in the process of aging. This I will not talk about much over here at this stage, but reactive oxygen species are known to damage a number of cellular components. So, how do these reactive oxygen species increase in our body? It gets increased when our body is exposed to certain toxicants. Like, for example, if a person is constantly drinking a lot of alcohol, or if he's constantly doing drugs, these substances do not find any place in our body. That is, they are not beneficial to our body. And as a result, your body tries to detoxify them as much as they can, as fast as they can. And in the process, one byproduct that is being created is these are these ROS, or reactive oxygen species. The number of other metabolites which are also created, but ROS are also produced in the process. Now, ROS uh, will try to be detoxified by the antioxidant system that is there in our bodies. So the antioxidant systems which are there, like catalase, superoxide dismutase, glutathione peroxidase, they try to reduce the levels of these free radicals. They get converted into some non-toxic forms like water, and then they can be excreted out of the body via sweat or via urine. But obviously, our body has only a limit as to how much antioxidants we can produce, these enzymes. We can also take antioxidants from the food that we eat, like from certain fruits and vegetables. But if the level of ROS in our body is too high, it will already cause some type of permanent damage to our cells. Okay, and if it damages the DNA, you can say that it can lead to a mutagenic condition, which can then further progress to a carcinogenic state as well. So ROS has been implicated as the mechanism of DNA damage in a number of chemicals, particularly the heavy metals, pesticides, and even microplastic pollution is said to be the toxicity that it exerts is via the production of ROS. Okay, so you can see the oxidative radicals that are there. They can actually convert the bases into different forms, and that can cause mispairing to take place. It can also lead to DNA strand breaks. It can lead to cross-linking also. Okay, so a number of things can occur because of ROS. Yeah, so you can see DNA strand breaks, which are basically the the a fragmentation or a break which occurs in the sugar phosphate backbone. You can see a break which has occurred. It can occur on either one strand, which is referred to as a single strand break, or it can occur on two strands at the same place. On both the strands, it is referred to as a double strand break. So there are a number of compounds which can also do this, which can cause breaking of the DNA, and that can then further lead to fragmentation of the DNA. If the DNA is going to be fragmented in this way, you can see that if there is a gene that is there on this stretch of DNA, it would not be properly transcribed. And if that gene is not going to be transcribed, we would lose that information of that particular protein, and that protein would not be able to do its particular function in the body. So from DNA damage, we move to the chromosomal damage, and chromosomal damage occurs because of DNA damage only. So the DNA strand breaks which take place can lead to, on a larger scale, can lead to chromosomal breaks. As you can see, breaks which are occurring in the arms of the chromosomes. Okay. Now, if there is a gene that is traversing that particular section of DNA, it would not be able to be transcribed properly. So when such conditions occur, most of the times, if there's too much of damaged DNA, the cell usually undergoes apoptosis, that is a programmed cell death. But sometimes the cells can even escape this mechanism, and then they can get converted into a mutagenic state and into a carcinogenic state further on. Yeah, so the most severe type of these breaks can lead to fragmentation of the DNA. So you can see how the chromosomes are getting fragmented. Okay, fragmented DNA is usually a sign for the cell to undergo apoptosis. And in most cases, fragmented DNA does not get propagated into the daughter cells. But there could be certain instances where the cell can escape this apoptosis mechanism, and it can get propagated into daughter cells also. But usually, because there's a loss of genetic information, complete loss of genetic information by fragmentation, usually the cells don't propagate further than this. Then we can have deletions. So deletions are basically portions of the chromosome which are deleted, as we can say. So a portion of the chromosome gets broken and it gets degraded or deteriorated. We refer to it as deleted. And then whatever remains of the chromosome get fused back together, and you can see that the chromosome has become a much smaller size now. So deletions are implicated in a number of genetic diseases, and this mostly you can mostly find this happening during the process of meiosis. Okay, and there are a number of genetic diseases which are known, which are caused because of deletions of the chromosomes. So there are certain compounds which can also cause these breaks to happen, obviously, and that can cause portions of the DNA to become deleted. Then we have chromosomal translocation, which is basically the exchange of segments of different chromosomes, again brought about by breaking of the chromosome. And because these broken fragments of the chromosomes are unstable, right, they try to bind with something to regain its stability. Okay, so we can have two different types of translocation. That is reciprocal translocation, where fragments of two chromosomes are going to be exchanged, and we can have another type, that is the centric fusion, in which two telocentric or acrocentric chromosomes can fuse with each other to form a very large chromosome that looks like this. That is referred to as centric fusion. And then we can even have a Robertsonian translocation, which is where two chromosomes they break at certain points and they form a very large chromosome or very small chromosomes. Then we can have inversions also, which are again involving breaks of the chromosomes that causes the chromosomal portion not to get degraded but to be inverted. Okay, so inversion can take place if it happens on one arm of the chromosome. It is referred to as a paracentric inversion. If it involves both the arms of the chromosomes, it is referred to as a pericentric inversion. So what happens in this case is that the genetic information which is there becomes altered, in the sense that suppose there is a gene which is traversing this region of the chromosome over here. If it gets inverted, the information is not complete. The gene which is there would not be, it's not a complete sequence of the gene which is there. And although the information is retained, but it is not a continuous stretch of the DNA, which is going to be probably an important gene which is there. So as a result, it would not be transcribing into its mRNA, and that mRNA would probably never be translated into a protein. Okay, so this is there are chemicals which can do this. So this is the inversions, the paracentric inversions and the pericentric inversions. We can even have dicentric chromosomes that can form, which are basically where chromosomes break at certain points and the fusion can take place. And it looks like as if this particular chromosome is having two centromeres, but it is actually brought about because of breaking of two chromosomes, probably during the process of meiosis or even mitosis, and that causes these fragments of the chromosomes to become unstable. And just remember that when chromosomes are unstable, they try to bind with any other structure within the nucleus. Okay, so they can either bind with other chromosomal fragments, as in this case over here, where it can lead to the formation of two centromeres, or they can fuse upon themselves like this, where the telomeric region of the chromosome gets deleted, and then the chromosome becomes unstable, tries to regain its stability, so it fuses with its own self, and you can see then it looks like something like this, which we refer to as a ring chromosome. Okay, so there are a number of chemicals which can also cause problems in the telomere, that it affects the telomere stability, and therefore it will affect the chromosomal stability, and then it can cause the fusion of the chromosome upon itself like this. Thank you.